Clown. All right, guys. So I think today we're ready to go on our next exciting adventure in biochemistry, and I hope everybody is getting seriously stoked for the new exciting installment we're going to be covering today. I'm trying to get my chat going here too. Where's my chat? There we go. Got the chat. Sounds like a great brand name. What I really appreciate about this class is it's got a good sense of humor. All right, so today we're going to talk about growth laws. Woohoo! Growth laws, growth laws. So we've been talking a lot about how organisms transform things that we eat into biomass and use it for energy. But what we haven't talked a lot about what limits our ability to do that. So let's get into it. So without further ado, one of the things I first want to talk about is what exactly are you, right? What in chat, like you've probably had this conversation, but what makes up a person, right? Well, about half of you, if you go by number of cells, is bacteria. Yeah, if we're going by total number of cells. So you are true. You do have a lot of water. That's right. So Robin's right there. But if you're going to talk about living composition or cell wise, like about half of you is bacteria. You're a giant vehicle for moving around microbiome, basically. And then if you break that down in terms of what your eukaryotic portion of you is, you are mostly red blood cells. So and so this is this distribution here you see up on the top, where we have this microbiome is this is your microbiome here, and then this is in terms of number of cells, like you're mostly red blood cells here, and everything else sort of falls into this category. So I don't know if you find that surprising, but most of you is bacteria and red blood cells. Now, if we take a different view and say, okay, what are you made out of by mass? Well, then the view is a little different. Like then, you're only 3% by mass by mass red cells, and then the most of you is made up of fat and muscle. Not too surprisingly, with other being everything else. This, of course, assuming like dry weight distributions that we're talking about. So that's interesting, but this kind of the high level view. How do we actually want to do that? Hey, where's the molecular composition? This one here. Now let's talk about the molecular composition of you. What do you think you are? So this is a distribution of composition of E. coli, which is say a regular representative cell. And if you're going to break down all the macromolecular composition of this cell, what do you think? What do you think it would be? So here's the distribution. Let's find the labeling. Carbon. Okay, so that's the atomic composition. So I'm looking for the macromolecular composition. Peptidoglycan. Okay, so let's see. We have peptidoglycan. Glycan. Okay, so there's one. Amino acids. Whoops, go back. Stop that. We have amino acids. Okay, what else? Chromosomes, plasmids. Let's just call that DNA. Okay, lipids. Okay, metabolites. All right. What else do we have? Carbohydrates. Okay, what else do we have? Is there anything else in a cell? Ribosomes. We can call that proteins. Okay. Okay. What else? Anything else? We got DNA. How about RNA? Okay, we can talk about RNA. Is there anything else that goes into a cell? Yeah. Okay. So let me just now condense your list here a little bit and say that this is metabolite slash amino acids. So that way we can ignore this portion of it there. And proteins, peptidoglycans, carbs, ribosomes. Ribosomes, we can, ribosomes are part of the protein and RNA. So we'll just say that this is RNA, DNA, lipids. So this just look like a sort of, yeah, there's some dissolved gases. Okay. So like dissolved CO2, O2. Okay. Now let's assign some percentages here. So what do you think? So this 55% here, OMP, proteins, in your opinion, would be 60%. So what do you think? So 55% here. That's one of the percentages. So what do you think of these classes I have listed here? Which one is the 55%? We've got a protein. What else? Any other votes? Metabolites. We have a vote for metabolites. Cell wall, which would be peptidoglycan, RNA. So DNA. Okay, so we have lots of guesses. Let's do a poll here. So I think that we've gotten a few guesses here. So I'll give you some options here. We have vote for, let's make this 1. What did we say? DNA, RNA, 2, 3, and 4. Okay, everybody type their votes into the chat. 1, 2, 3, or 4 for the 55% level. Yeah, so we have an overwhelming favorite for this being protein, and you guys are right. Well, hey, stop that. Stop that. This here is in fact protein. For goodness sake. This I'm trying to do this on my Surface. It's a lot less key right here. Why don't you just go away? Keep them. All right, here we go. So hey. Actually, it might be easier if I type it since I'm on the Surface here. So let's see, right here, this guy is in fact, that one is in fact protein. So, all right, now the next one, what is this 20%? So let's have our votes, 1, 2, 3, or 4, or is it something else? 5 being something else. 3 votes in the chat. What's this 20%? 5? 5 being something else. 5, 5, 3, 3, 4, 5, 3, 3. Seems like we have a split between 3. Anything else? 4 metabolites. 5 being all other things. Hello. 5 being other. 5 would be carbs, gas, lipids, peptidoglycan, any of those. What do you think? So let's see it again. Votes now. 1, 2, 3, 4, 5. Obviously, it can't be 1 anymore. So we'll have this be 1. Sorry. All right. Now we got it. 1 being peptidoglycan, 2 being DNA, 3 being RNA, 4 being metabolites, 5 being other. What do we have? 3, 4, 1. I'm going to just assign all these now. 1, 2, Hey, go back. 5. So we have quite a mixture of opinions here. So let me assign some of these other ones here. And Carly, you may have to help because I don't have my notes with me here. Make sure I get this right. But this guy right here, this little fraction, I think this one is lipids. Is that right, Carly?
Yep, 9% is lipids.
Yeah, lipids. Okay. And this guy here, this guy is DNA. This guy here is metabolite levels or something of that nature, carbohydrates, metabolites. Gases are out at the, fraction of a percent, tiny portion of it. So what the heck is this 20% of the cell? This is a question to the class, not to you, Carly.
Okay.
I know it.
Also, the key is in the notes, by the way.
Oh, it is. Oh, shit. Really? Yeah. Oh, I see that. So those of you who are reading the notes, very good. So this is RNA, right? Stealing my own thunder. Next time, we got to remove the notes there. But this is RNA, and if you find this surprising, it should be like 20% of your cell is RNA. And so, like, if we're going to make a breakdown of that, what would it be? Is this you have the options here of one is message, mRNA. B is tRNA. And C is rRNA. So what is 1, 2, 3? What's making up most of that RNA? 1. We have votes for 1. Lots of votes for 1. 1, 1. Yeah, unanimous votes for 1. So you guys are all wrong. mRNA is like down here in the fraction of a percent range. Okay, now we have votes for 2. How many votes for 2? All right, yeah, now you guys have got it. It's actually ribosomal RNA. So 20% of the cell is RNA, 55% of it's protein, 10% of it's lipids. Everything else is the remainder. So what are you? Is this wild? What are you? Well, let's go back to that original thing where we started. You are bacteria and red cells, if you want to take a cellular view. If you want to take the mass view, you are fat and muscle. And if you want to take the molecular view, you are protein, at least on the cell component, you're protein and RNA. Weird. Okay, biochemist, are you feeling is this a mind-blowing experience? Yeah. Is Stoke high? Well, I've just told you what you're made out of. You're like, what am I? You are a bag of bacteria and RNA. Well, of course, this sort of high RNA component is a description of, that's a description of, free living cells, right? So is this incomparable in eukaryotes? Yeah, you see some things like this in eukaryotes. Now, these percentages, as we're going to talk about more, are quite variable, and they depend an awful lot on the cell. And there's some major exceptions to this. Like, for example, E. coli grown in osmotic conditions can concentrate half their mass as glycine betaine as an osmoprotectant. Do you ever feel like a plastic bag? I do all the time. A bag of Stoke. So there are other organisms that can accumulate lipids in them to a significant fraction of the dry weight, and there are other kinds of molecules that can be accumulated to very high levels. But the thing I want to bring you back to is now, so this composition, like what we are, has a really cool and like a significant insight that it gives you into what it takes that limits our ability to grow. So let's go back and talk about microbes growing in culture here for a little bit. We have, this is something that you guys have all seen before. So what's going on here for somebody who's had microbiology class? Like, give an example of this. What the heck is this thing? we have some unfocused chatter here in the chat. What are you guys talking about? You feel like paper thin, like a house of cards? We're talking about your composition. But what is this? What is this figure here we're looking at? What is a growth curve? Yeah, like this is classic biochemistry, microbiology growth curve. You see the bacteria grow, they get to some phase. What prevents them from growing? Why don't they continue to grow forever? Why doesn't the curve? This is really obnoxious. My little pen isn't really doing what its thing. Why doesn't it continue to do that? Nutrient limitation, obviously, right? So that's what divides up these standard phases. So we have lag phase where they're getting going, and you have log phase where they, they're in their max growth, and then you have this sort of declining phase. And you have this eventually, stationary phase, and then you have this death, right? Like this is classic microbiology. But here's something I'm not sure this has come up before in your class. What limits this growth rate there? Why can't it grow faster? E. coli growing in culture, why can't it be like that? What limits its maximum growth rate? Right? So it makes sense that the most it can grow to here is nutrients, but why can't this, the derivative of this slope here, be higher than that? Speed of DNA replication, there's some guesses, synthetic rates, ability to process food, lots of good ideas. Let's doubling rate. Yeah, but what is it that makes the doubling rate? So E. coli grown in culture is 20-minute doubling time. Why isn't it 10 minutes? Why isn't it 3 minutes? Right? Time for cells to divide, right? It's an interesting question, right? E. coli has evolved to be one of the fastest replicating things on the planet. And there are two basic strategies for living. One is to take care of your babies and make sure that you invest lots of energy in ensuring that that your babies have babies, right? That's one version of it. Humans are an extreme example of this, where we maintain our babies here for like 20 years. And then the other version of this is E. coli, which is let's make babies fast. So that's a good strategy when you're living in a really abundant nutrient source and you want to make babies really quickly. So E. coli is extremely well evolved for that particular style of replication. So what is it that determines its growth rate? Let's talk a little bit more about this. So classic microbiology says that the growth rate of a microbe can be predicted from the concentration of some limiting nutrient, some substrate concentration, and that you can use this limiting nutrient to characterize the growth potential. And this has this equation here that says that mu, or lambda, frequently is lambda, but or mu, the growth rate is at some particular concentration of nutrients is a function of the maximum growth rate multiplied by the substrate concentration divided by an empirical constant, which describes the sort of the nutritional accessibility, effectively the nutrient plus the concentration of the substrate. Does this look like a familiar curve? Has anybody seen this curve before? Michaelis-Menten, yeah, exactly. It's the same curve. There's nothing really magical about that. All that says is that this particular curve describes a line with this shape. That's all that curve says, right? But it's a useful tool for describing that kind of curve. And so microbes have this kind of behavior when they're exposed to varying concentrations of some limiting nutrient. They have a growth curve pattern that looks just sort of like Michaelis-Menten, right? So as we said, like these are the compositions of the cell. Whoa, protein! We should do some proofing here, but evidently it's in the chat or in the notes here is laid out in the percentages. But as we just got finished saying, so all right, for your rapidly dividing generic cell, it's like roughly 60% protein, roughly 20% RNA. Right? All right. So we're still working on this question of what limits microbial growth rates. Why can't they go faster? Okay. Well, as it turns out, There's a beautiful paper that was published in Science. Carly's put this in the chat. That was exploring the interdependence of growth rates on these factors here, and it's a really great example of some mathematical predictions that ended up being used and tested in the real world. It's a beautiful paper. I love this thing. So I encourage all of you to read this. It's science at its very best, in my opinion. So let's talk a little bit about the observations that went into this study. So first off, one of their observations was that, when you grew microbes in different media that had higher and higher nutrient contents, their growth rates were higher. So this is like your classic curves of, hey, stop that. This is your optical density. So this is the total number of microbes per mil. And this is, for each of these guys, we have improving nutritional quality here. So like each of these media have better nutritional quality. I don't know why my pen is so glitchy here, but it is. So where this is very high quality media and this is very low quality media, and as a nutritional composition of the media improves, you get faster and faster growth rates, as is evidence from the difference in these slopes, right? You see that? The derivative here of the OD curve is the growth rate. Does that make sense to everybody? Is everybody tracking so far? Better nutrients equals faster growth. Yeah? Yeah? Does that make sense to everybody so far? Chat? Yes. Okay. So I can take that same curve and I can plot it on a different axis here. And this axis looks like where we put our growth rate, which is, so this is that, this pen is really crummy. So this growth rate here is the derivative of the, this is the derivative of the curve here I was showing you just a second ago. And again, what I'm showing you is nutrients of improving quality here along this axis. And not surprisingly, as we move along this axis, the growth rate improves. Yeah. So the other thing, though, that they showed that was really cool is the RNA content of the cells also increased over time. So as the growth rate went up, the RNA content of the cell changed. And so that's pretty interesting. And that was mostly made up as the RNA that's built in the ribosomal fraction here. So this is a complicated curve that says, like, as growth rate goes up, more and more of the cells are made out of ribosomes, right? And they follow this linear equation here, where there's some maximum amount of RNA is a function of some total minimum content that's possible, plus the empirical growth rate here, lambda divided, or the growth rate divided by some empirical constant, which is called kappa t. Yeah. So is everybody following so far? The growth rate is linearly related to the RNA content, and it follows this linear equation. Does everybody track so far? If you know the growth rate, you know the RNA content, and it follows a linear relationship. This should be pretty wild here. Like, I could tell your biomass composition by how fast you make babies. That's pretty cool. So then they did another experiment. All right. In this experiment, they used a tool called chloramphenicol. So let me have a little sidebar. This is my tangent cam on antibiotics and chloramphenicol. So many antibiotics target the ribosome and target translation. Okay, antibiotics, this is a convenient feature because eukaryotes have fairly different ribosome structure, and so we can make antibiotics that target the microbial ribosomal or translational complexes, and they're safely different from our complexes, so they don't interfere with our translation. So this is a good target. So this is a slide of all kinds of different antibiotics that target different functions of translation in microbes. But chloramphenicol, which is the one we're talking about today, is this. It's like there's this structure right here, and chloramphenicol actually binds to the 50S subunit, inhibits the formation of a peptide bond. So it actively interferes with the translational process. Okay, load that into RAM. We're going into our next slide. So the next experiment that they did is they say, okay, well, we already showed that increasing growth or increasing nutritional qualities gave higher amounts of RNA content. And so you can imagine here on this plot, this thing is really buggy. You know, as we move along this direction with better and better nutrients, they get you know, faster and faster growth rates. So the question they were asking is, what happens if this goes on to infinitely good nutrition? Okay, like, you know, infinitely good nutrition. Well, will this continue on into infinity? Like, can you get a growth medium that doubles the growth of E. coli beyond what they have and say, you know, yeast extract? What do you think, chat? Is it possible to get a better growth rate than you know, what you get in your very best microbial growth media? No. We're having votes for no. Yeah, so basically our microbial growth media, rich microbial growth media, have already been optimized to get essentially as good a growth rate as possible. So everything is a function of that. And this guy is, you know, maybe not perfectly at the max, but very close to, you know, is an optimal growth medium. And so the question they were asking is, so what is the ribosomal composition if this were taken to absolute perfection? What is the total maximum RNA content that could be in a cell, right? If nutrient quality predicts RNA content, and that predicts growth rate, and what is the maximum amount of ribosomes that you could have in a cell, right? So what they did is they used that chloramphenicol drug at different concentrations. And the idea here is that by inhibiting protein translation, you can prevent the biosynthetic rates artificially and find out by titrating them in. You can titrate in and find out what the maximum ribosomal content is possible for a cell, right? So they found that these things all have this sort of, you know, as you titrate in different concentrations of chloramphenicol, Microsoft Surface is not a replacement for an iPad. Is that, you know, as you go that way, the you know, adding in higher and higher concentrations of chloramphenicol, you ultimately regress to some particular maximum, in this case 0.8 RNA mgs of RNA per protein, and it didn't really matter what the nutrient quality was. They all sort of regress with different slopes to that same intercept, right? So what that told them is that the ribosomal content also followed a linear equation, which is that the ribosomal content for any particular, at any particular time was a function of the maximum RNA minus the growth rate divided by some empirical constant, translational constant here, which is kappa n. Yeah. So they also found in this thing, so in E. coli, that the maximum RNA content here and then is 0.55, something like that. Interesting, yeah? Why does chloramphenicol concentration lead to increased RNA content? Well, so this experiment's a little counterintuitive. So what they're trying to do is they're trying to mimic infinite nutrition, right? So ultimately, that's what they're trying to do. They're trying to find out if you take this, you know, this trend that we're observing to extreme, what is the total amount of RNA possible inside the cell, right? And so the way they're mimicking that is they're preventing the cells from making protein, right? So in that way, you can find out if the cells were only made out of RNA, or what's the maximum total RNA that they can have, right? Does that make some sense? So they're this is bringing them to their extreme. This is an empirical experiment, like trying to establish that, right? Is it an awesome a substitute for this maximum nutrition kind of experiment? I don't know, but it's a reasonable approximation of it. Yeah. Okay. So what the heck is going on here, right? Am I the only person who's wondering this? We're talking about linear relationships between growth rate and RNA and biomass composition. What the hell is the point? Right. This is the conversation spaces of people in chat, right? So let me explain. The cell its composition is what this says is a few fundamental things. One is that the biomass composition of the cell is not fixed. It changes with respect to the nutrients in its environment. And so as the nutrients are better, the cells grow better, and a greater fraction of the cell is made up of ribosomes, this RNA portion right there. And when cells are in a lower nutrient environment, a bigger fraction of their RNA is made up of protein. And so let's think about this intuitively a little bit. So when you're in a very, very rich environment, you're living at the supermarket, you don't actually have to walk very far to go and find your favorite foods. If you want to only eat at the cake counter, then you can just stand at the cake counter and eat cakes, and which means you don't have to walk around to the flour and the baking soda and the sugar and all that and bake a cake. That takes a lot of work. Well, the cells are kind of the same. Like when you have access to outstanding resources like glutamine and glucose, like there's not a heck of a lot of reason to have transcription of all kinds of metabolic pathways on that allow you to, say, catabolize galactose or whatever, like taking an example from lycopteron. Um, um, so when you're in nutrient-rich conditions, the cells can lower the amount of protein that is devoted to things other than making new cells, and they can devote most of their biomass to replicating quickly. In that case, they optimize themselves for ribosomal content. Okay. And when they're in nutrient limited conditions, the cells have to invest a lot of energy into getting their biomolecules in. That means they need to make more proteins. They need to invest more energy into each of the nutrients that they bring in to get it into the right form. And as a result, they have a bigger fraction of their proteome is made up of these non-ribosomal associated proteins. Does that make sense to everybody so far? Faster growth, better nutrition, higher ribosomal content. Yeah? Yeah, from everybody? So this tells you something profound about biology, though. This isn't just observations about how cells change. It tells you something profound about how cells divide, right? It says that what limits a bacteria's ability to grow, why E. coli can't grow faster, is its translational capacity, right? Why do you add more cells, or why do you add more ribosome? The reason why is that is the rate limiting step in making a new cell. So by adding more and more and more ribosome, you increase that rate linearly with respect to making, linearly with respect to ribosomal content, right? And so when you're in nutrient poor conditions, every protein that you have to make that is not a ribosome steals from your translational capacity and lowers your growth rate. Does that make sense? Does that observation make sense, chat? So what limits microbial growth? Why can't it grow infinitely fast? Yes. What is it that limits microbial growth? Translational capacity. You're right on the money there, Jeremiah. Ribosomes. Yeah, Jared, you're right. This is the translational capacity that's made possible by having those ribosomes. So this observation one, it tells you, to me, is just a like, it's really profound kind of insight into what one of the fundamental speed limits in biology. And the other thing it tells you is it has some really important practical implications. So let's talk a little bit about that. So one of the things that they showed there is that so the growth rate that's possible can then be modeled very neatly by this equation. It says that the growth rate lambda at some, it's a function of two empirical constants here, this translational capacity and this nutritional accessibility. Those are both sort of empirical constants here. So these guys, kappa, these are just things you measure, right? And then so the growth rate that you actually observe, lambda is a function of some maximal growth rate, lambda c, multiplied by the translational capacity of the system, times the nutritional capacity divided by the sum of the two empirical constants, right? So in other words, it looks an awful lot like Michaelis-Menten. pretty interesting, yeah. That life's fundamental speed limit can be modeled here by a fairly simple equation, yeah. So let's talk about some of the practical implications of this. Have you ever heard of insulin? Like, where does insulin come from? Pigs. Originally, it did come from pigs. So it was originally it was actually isolated from the pancreas of pigs. But Tarek is right. It comes from bacteria now. Because getting it out of pigs involves growing a pig, taking it out, purifying it, and the pancreas, the beta cell in a pig is maybe I don't know, some tiny amount of material, right, out of this giant animal. So it's a pretty inefficient way to get it, right? So instead, what we've discovered is, hey, if you need a protein, the best way to make that protein is to encode it on a plasmid and put that gene into a bacterial expression system, grow the bacteria up, turn on the expression of it, and then you'll get your favorite protein, right? This is how molecular biology works nowadays, right? And so a big chunk of molecular biology and traditional biochemistry is making these proteins, purifying these proteins, and using them for things. And insulin's a great example of something that's our ability to do that has radically reduced the cost and improved the accessibility of insulin so that people can manage their diabetes now, right? So that's one really good example of something that comes from those kind of recombinant expression systems. But if you ever work with a recombinant expression system, something weird happens. So you put your favorite gene on there, and normally, you can have this thing under an inducible promoter like IPTG. There are these inducible expression systems where you can put in something like IPTG, and then the bacteria will turn on the expression of your plasmid. And so what happens though, if you're ever working in a lab that does this, is that after you add the molecule that turns on expression of your plasmid, your inducer, after induction, the cells stop growing, right? As we're showing here on this curve, the the number of cells effectively stops, and you can't get any more cells, right? When they start making protein, your favorite protein, but they don't make any more cells, right? So why does this happen? What's the reason for that? Now knowing what you know about Hua and growth flaws, yeah, they're making proteins instead of ribosomes, right? So Jared's talking about toxicity and this sorts of things. So yeah, I mean, there are other reasons why it can happen, but the point from this lecture is that one of the fundamental reasons why they're doing this is that you're stealing from their translational capacity. Like when you turn on expression, particularly, you normally turn it on with a really heavy duty promoter that effectively redirects all of the translational activities to the cell to this favorite protein. What you do is like they're not making other stuff anymore, right? And so there was a beautiful experiment by Waugh in this follow-up where, so he did this empirically. So what he did is he took a protein, in this case it wasn't insulin, he put beta-galactosidase on this inducible promoter system. And so he put this under the control of some transcriptional factors that had a big range of recruitment ability. So like the strength of expression was different, or the magnitude of expression was different. And what he showed was that the growth rate in the cells was inversely proportional to the expression rate of this protein. And so you could actually model this here again linearly with this system where the growth rate here of the cell, darn it, come on, is inversely proportional to U. You can call this U, right? So we can also write this out as an equation where we'd say that, so if you were to assume that the proteome, this is the total proteome is a can be represented as a function of Q, if my pen will ever write, Q, which is a housekeeping protein, minus P plus R plus U, right? Or I'm sorry, P plus R, this should be a plus. P plus R plus U, that makes sense. So that's the total fraction. So if you were to rearrange this and say that then, so this total fraction of the proteome is made up of all these pieces, you can say that, as U increases, if this thing all equals 1, say if this all equals 1, as U gets bigger, something else has to get smaller for this to equal 1, right? And in this particular case, Q is a housekeeping protein that can't change, like actin or something like that. So the only thing that can go down is your other fractions here. And so these guys get smaller. And so as a result, growth rate goes down. Does that make sense? Pretty neat. Does that make sense, everybody? All right. So what the heck does this mean about biology? So there's some really cool insights from this in biology. So Pseudomonas, let's talk about Pseudomonas aeruginosa. It's a microbe that lives in soil, lives in drains, it's an environmental microbe, lives everywhere. But the one place that it's funny is it can also live in the lungs. And it lives in the lungs of people who have cystic fibrosis. It forms these long-term infections in the lungs. It can also happen in people who are intubated. And so the thing that's interesting about these infections is that they're like, these infections can persist for 30 years. Like people with cystic fibrosis will, can have these infections for a very, very long time. And so the microbes that are colonizing them also can evolve over a very long period of time. 30 years is an extremely long evolutionary history for a microbe that's got a doubling time of an hour, right? So something that's peculiar about Pseudomonas that's isolated from lungs, 40% of isolates are auxotrophic. So auxotrophy, to remind you, is that these microbes require exogenous sources of amino acids in order to live. So they're auxotrophic for arginine. 40% are arginine, oxytrose, and other things. So they start to become really dependent on exogenous sources of amino acids. So why the heck is that happening, right? Well, I want to pose this back to you as a thought experiment. Now, what you know about this growth raw data that we're just talking about. When you're living in the lungs of somebody, or let's say you're a pathogen living in the bloodstream, you have infinite access to amino acids. So my question to you is, would it be in your evolutionary advantage to lose the biosynthetic capacity to make those amino acids de novo if you always had an exogenous supply? What do you think? In other words, if you were to reframe um, this whole question, you go back and you say, okay, here's your portion of the proteome. There's Q. This is the fraction that we can't change. This is the housekeeping stuff. And you have some portion P, which is proteins, and you have some portion that's ribosomal. So what's going on here? It's easier to steal than to make. Yeah, Jeremiah's right on the money. And so that's exactly kind of what I think is going on. So this is speculation or conjecture or theory on my part, not proven fact. But this is the cool, fun edge of biochemistry. So what I think is going on here? Why are all these Pseudomonas auxotrophs evolving? I think what's happening is, as they live for a long period of time in an environment where they have infinite access to amino acids, some of the biosynthetic pathways ultimately move from the P category into the U category, and they just become unnecessary. And so by dropping these guys out, you actually get a wider and wider fraction of ribosomes that can be made and used for either making new babies to outgrow your competitors, or combat the immune system, or a variety of other things, right? So to summarize that, I think that the access to nutrition has a really profound impact on what things are necessary or not necessary. And if you hold them for a long enough period of time, some things can be evolved away, right? It's nutritional selection. Is that interesting? You guys like that theory? Help you understand the evolution of microbes. So here's to leave off with. Actually, I guess we're just about on time here. So actually, I'm gonna just make this one very. I'm going to just not even bother to cover this one here, other than to say that we're studying these systems already in our laboratory, and recently we've found that that Pseudomonas, the same microbe we were just talking about, has a really interesting safety valve that it's evolved to deal with changes in nutritional composition. And so here's an example where we've grown Pseudomonas strains from a variety of different nutritional conditions, from bad to really good nutritional conditions. And we've also looked at a variety of Pseudomonas isolates from people's lungs with cystic fibrosis. And we've shown that the total amount of biomass they produce, or this, something proportional lambda that we're talking about, is directly related to their carbon commitment. Well, how much carbon that comes into the cells and they commit to making biomass. So these two things are linearly related in that the nutritional composition predicts this. So this is following along from the WA predictions. Well, as it turns out, and we don't have time to give you the whole story, one of the things that's going on here in Pseudomonas at the molecular level, it's a real world example of the shift between the P fraction and the U fraction we're talking about, is that when Pseudomonas is grown in these very nutrient limiting conditions, it's evolved a way to convert glucose into 2-ketogluconate and gluconate and use that to fuel its electron transport via this periplasmic oxidation of glucose without ever committing carbon into the cell. In doing that is a really extreme example of this, this selection for, or turning down production, unnecessary protein production down. And so what it does in this particular case is it uses this all completely periplasmic mechanism for getting its energy, and in so doing doesn't have to commit any carbon inside the cell. Anyway, it's a first little foray into this idea of how nutritional selection influences, or nutritional environment influences the composition of cells, and how the biochemistry behind this interaction between growth rate, ribosomal composition, and survival under either nutrient fit or nutrient poor conditions. Anyway, that's a summary of what we want to do today. I'm happy to hang out and chat if anybody has any other questions.
Dr. Lewis.
Yeah.
Have you guys done any like research into, because when I was working in the Harrison lab and I was doing an independent research project there, I was working on cystic fibrosis. I was just wondering, like, have you guys